Assalamu alaikum students. Today we'll study one of the most interesting and informative topic, that is cloning. What is cloning actually? You can simply say that a cell or individual with its asexually produced offsprings. Now over here our word is asexual. What does it mean? Asexual meaning is that the type of reproduction that doesn't involve the combination of the female and male gamete. Then there comes that why we use cloning, for which purpose. The reason is that we use it for eugenetic aims. This eugenetic, it has come from the Greek word eugenes, which meaning is good breed. Then eugenetic aims are basically related to the healthy offsprings or to the better offsprings. For this purpose, cloning is used to form an exact duplicate of the individual from which the cell is taken. Now we will study further about is that which sort of purpose is included in which sort of purpose of that in this sort of method we can do cloning, and of which things we have done the cloning. But first of all, we'll go towards its historical background. First of all, cloning was tested and performed. It was experimentally performed by a scientist of Scotland in 1997, and the first experiment was performed on a sheep. In that case, the name of the sheep was Jolly. Now I'll tell you the procedure that how we can perform cloning. And also remember this thing that up till now the most of the organisms which are used for cloning are mice and cows, because these are very useful animals and used for various purposes. Okay, now let's discuss about the procedure through which we can do cloning. Let's take the example of a sheep. This is a sheep. For example, I take a female sheep. I'm interested in that. Then female sheep I've taken, and it is very useful for any purpose. That's why I want a clone of it. What I'll do, that the cell, or you can say the fertilized egg which is present in the womb of the sheep, I'll take that out, and over there will be a nucleus. It is fertilized egg. Now I will take out a cell from the, suppose it is a sheep, then I'll take out a cell from its skin cell, because the skin cell is fully developed by the female sheep. Now suppose this is the somatic cell, you can call it, or you don't have to use any that sexual cell. You have to always use somatic cell for this purpose. Remember this. Now somatic cell I have taken from the skin. Now I'll take its nucleus. First of all, I'll discard this one nucleus, and this thing we have to perform at micro level and at microscopic level. We have to perform this, and a slightest mistake can spoil all the procedure. So we have to be very conscious about this thing, that we are performing each and everything totally perfect. Now, as I've removed this one nucleus, and I will now take out this nucleus and I'll put in it. I'll take out the nucleus from the somatic cell and I'll put in the fertilized egg, which is not containing the nucleus, from which nucleus has been discarded. Now this new cell will called alternated zygote. Now I'll take this cell and I'll put it in the womb, or you can say uterus, of the female sheep, and then the natural process will continue, that it will develop into a new offspring that will be the exact duplicate of the organism which nucleus was put in it. The nucleus which was changed in the nucleus which was present in altered zygote of that organism, it will be the exact duplicate. There will be the 100%. They will have 100% similar genetic as well as physical appearances. Physical and genetical traits both will be same, 100% same. Then there's one more sort of cloning. Let's discuss that as well. The cloning which we have discussed earlier, that doesn't take place naturally, but this one cloning do take place naturally. That in this sort of cloning, what we do is that in this sort of cloning, the both organisms, the both offspring will be similar to each other, not to the parents, but they will be the exact duplicate of each other. In this, I'll take a zygote and I'll provide it such conditions that it will do mitosis, and through mitosis it will divide into two zygotes, two daughter zygotes. Then I'll put both of the two daughter zygote into the worm, and don't forget that they will be totally similar in the structure because the deviant was mitosis. Now I'll put both of them in the worm of the female, then they will continue the natural process, and the two duplicate 100% duplicate offspring will be formed. This is one more type of cloning, and this is a very famous question, that why we don't use the cloning for the human beings. Always remember that at some places scientists are making attempts to clone embryo of the human, which they believe can serve a transplant donor. It can be served as transplant donor. There's a lot of controversy on this issue because the human cloning should not be attempted or not. Why is that so? Because sometimes it happens that if we will create the clone of the human, it will create a lot of problems and a lot of troubles in the life. For example, if the clone do any sort of crime, then it will be difficult to adjust that either the clone or the real person has done the mistake. So it will be difficult to identify that. So to avoid such disturbances in the environment, we avoid this cloning. So that's all about the cloning. I hope you have understood it properly, and if you have any sort of questions, you can ask me in the comments below, and you can also like my video. You can subscribe my channel. Thank you for watching the video. Allah Hafiz.